Dear student, you are welcome again for the very important lecture in the molecular biology, which is called gene cloning. As you know, gene cloning is defined as the reproduction of DNA fragment. Reproduction of DNA fragment, and in this lecture we will talk about the main two processes of gene cloning, which is cell-based approach and PCR. I will give you briefly the basic informations regarding this lecture. The unit of heredity is the gene, and the gene is the segment of the DNA that carries in its nucleotide sequence information for a specific biochemical or physiological property. In all organisms, the genetic material is stored in DNA, with the exception of some RNA like QPMS2, some RNA virus like influenza virus, rotavirus. These viruses are preserved in the RNA ribonucleic acid. Okay? الجين يا طلابنا هو البasic biological unit of heredity, which is a segment of deoxyribonucleic acid needed to contribute to a function. When I talk on the unofficial definition of gene, according to the official guidelines for human gene nomenclature, a gene is defined as the DNA segment that contributes to phenotype or to genes are composed of DNA, a molecule in the memorable shape of double helix, a spiral ladder. Each rung of the spiral ladder consists of two paired chemical code bases. There are four nitrogen bases, as you know: adenine, thymine, cytosine, guanine, and each of which will start from the first letter of its name: A for adenine, T for thymine, C for cytosine, and G for the a gene is a sequence of bases. It's made of combination of adenine, thymine, cytosine, and guanine. And these unique combinations, هذه الاتحادات الفريدة, determine the gene's function, much as letter joined together to form word. الwords هنا نقصد بها الكودون. الكودون مثل ما نعرف, each codon consists of three nitrogen bases. Each person has the thousands of genes. For your information, the human being consists of 30,000 genes. Most of these genes are inactive or inert. Billions of base pairs of DNA or bits of information repeated in the nuclei of the human cells, which determine individual characteristic genetic traits. Genes are arranged in precise ways بطرق دقيق, يعني تواجدها مرتبة بشكل كلش دقيق ويونيك فريد all along the length of 23 pairs of much larger structures. مثل ما نعرف in the genetic, half of the genetic materials are inherited from the father, and the other 50% are inherited from the mother. Okay? The chromosome in any particular pairs look like each other, except in a boy, which is there is one pair of a chromosome, which usually settle the sex of the individual. This pair has two X, XX in female and XY in. Also, regarding the X and Y chromosomes, these chromosomes are always capitalized and are the sex chromosomes. All the other chromosomes in the human body اللي هي from 1 to 22 نسميها autosomal. They are called autosomal chromosomes or autosomes, while the letter pair اللي هو نسميه ال 23 نسميه ال 23rd ال 23 نسميه حقيقة نسميه ال sexual chromosome, which is responsible for gender. Now, as you know, the genetic elements in bacteria are different. Of these, plasmids, episomes, cosmids, transposons. Now, I will talk briefly on each of each one of these. As you know, plasmid. What is the difference between plasmid and episome? Plasmids are extra chromosomal pairs of DNA, which are lives, which are replicate independently of the DNA. يعني هو جزء مستقل خارج عن الكروموسوم يضاعف بشكل مستقل عن DNA, while episomes are integrated parts with DNA. It will replicate as well as DNA replicates. The spread of multiple antibiotic and drug resistance in pathogenic bacteria is one of the function of a plasmid. The second one is the instability of industrially important microorganisms. What are the type of plasmids? There are 5 types of plasmid. A fertility factor or fertility plasmid, which is responsible for fertility. A resistant plasmid or R plasmid, which is very important in gene cloning, and it is used as a vector, as we discuss later, carrying genes coding for antimicrobial resistance phenomenon. The third one is the coplasmid, which is coding for colicin, the protein that calls sensitive Escherichia coli to this amino acid. While the other type, Fibrio, from its name, it kills what? Sensitive Vibrio cholera. The fourth type is the virulence plasmid, which are responsible for virulency of microorganism. And the fifth type is the degradative plasmid, which is responsible for the degradation of tissue and bases at which the plasmid will located in it. Okay. All conjugative plasmid must have 2 characters. First, it must contain RTF, resistance transfer factor, and the other region is called R factor, R determinants, or R genes. Resistance transfer factor, it is responsible for transfer of resistance gene from bacteria to the another. Thus, no transferring from gene from bacteria to another in the absence of resistance transfer factor. لأنه بالأساس هو هذا نسميه بالعربية عامل نقل المقاومة اللي هو مسؤول عن نقل جينات المقاومة من بكتيريا إلى أخرى. The plasmid have transposon elements. Now we will talk on the third genetic elements, transposon elements or T elements, which also called jumping gene, which can pass from bacterial strains to another, and also between chromosome to plasmid, or plasmid to chromosome, or chromosome to chromosome by jumping movement, gene القفاز. Thus it's called jumping gene. What are the characters of transposon or TN elements? First, they are mobile segment of DNA, while they are motile. They are moved by jumping. They are perhaps it is mobile or motile DNA segment. Second, they contain genes coding for their rearrangement. يعني هذا الترانسبوزون من يجي يدخل إلى الترتيب وتسلسل القواعد النيتروجينية هيعمل تشينج بيها، فهو قادر على أن يعمل لنفسه rearrangement إعادة ترتيب. Third, they contain genes that specify resistance to various antibiotics. يحمل الجينات اللي هي عادة شفر للمقاومة. Now we will talk on the DNA recombination. We will enter to the core of this project اللي هو DNA recombination. Also it is called gene cloning. It is called also genetic engineering. All of them, it refers to the same term. Once the DNA is transferred from the donor to the recipient cell by one of the 3 processes, which are conjugation, transformation, transduction, it can integrate into the host cell chromosome by recombination. There are 2 types of recombination, homologous recombination and non-homologous recombination. In homologous recombination, there are 2 pieces of DNA that have extensive homologous region pair up and exchange piece by the process of breakage and reunion. While non-homologous recombination, it's rare and uncommon to occur. It needs homology for the Now, DNA cloning, gene cloning. What does mean by cloning? Is the reproduction of DNA fragments, as I talked in the first slide. Reproducing of DNA fragments. It can be achieved by one of 2 methods, either by previously by cell-based approach, and now after PCR invention, بعد اختراع PCR by Carey Mules in 1983, polymerase reaction was introduced in the practice to perform gene cloning. Now, in this lecture, we will concentrate on cell-based approach and followed by polymerase chain reaction. First, cell-based approach. In cell-based approach, a vector is required عامل ناقل نحتاج to carry the DNA fragment of interest in the host cells. And عندنا DNA fragments, I need to reproduce this DNA fragment. But I need a vector to carry this fragment into the cells. Thus, we'll show here the fourth essential steps of gene cloning: DNA recombination, transformation, selective amplification, and isolation of desired DNA clones. I will talk with each one in details. Now, regarding the first one, production of recombinant vector. Here, I have the DNA fragment to be reproduced, and this vector is needed to carry this fragment into the cells. This vector is called plasmid. Any plasmid to be conjugative, can make as a vector, it must have two characters. First, must be small in size, not large in size, because if it's large in size, it will be refractile easily. Second, it must contain two regions, R gene and RTF, as I mentioned previously. Now, DNA fragment with vector, it forms recombinant vector. And regarding this first step, I will talk in details. Here, this represents the plasmid, and this represents the DNA wanted to be reproduced. After using the restriction enzyme اللي هو اسمه EcoR1 مشتق من E. coli restriction enzyme 1. هذا كل انزيم قاطع إلى specific region for cutting. اوكي EcoR1 يقطع عادة من منطقة GC as you show here. It cut the plasmid and DNA from the specific region. يعني عمل الانزيم القاطع هنا not haphazardly مو عشوائيا، وانما يقطع من منطقه محدده. لذلك قطع من منطقه الجي سي منا بالتالي حصلنا شنو؟ حصلنا cutted pieces، قطع مقطعه او مقطوعه من البلازميد والدي ان اي. هنا بفعل الستيكنس اي الستيكي انس الموجوده في منطقه القطع للبلازميد وللدي ان اي. And due to presence of DNA ligase, this enzyme is very important for the hybridization or recombination. The sticky ends of the DNA will stick to the plasmid to form what? To form recombinant plasmid. This represents plasmid, and this piece of DNA wanted to be reproduced. Here I obtained the recombinant vector. Okay. Now, the second stage now is the how can complete the all other processes for successful DNA cloning, as I mentioned previously, but by other schematic representation. This is the plasmid, and this is the DNA to be wanted to be reproduced. And this plasmid, small in size, has R gene, which is resistance gene for antibiotic resistance. والexample اللي راح ناخذه بمحاضرة اليوم هو الجين اللي كأنتيبيوتيك هو الأموكسيلين. إذا هذا هو الجين اللي responsible for amoxicillin resistance. And this DNA wanted to be reproduced. After restriction by restriction enzyme, after cutting by restriction enzyme EcoR1, the sticky ends of both of DNA wanted to be reproduced, and the plasmid will be hybridized by hybridization in the presence of DNA ligase, will form recombinant vector, recombinant plasmid. The second stage is the transformation. In the transformation, this recombinant plasmid will transform directly through the cell wall to the bacteria. والبكتيريا المستخدمة هنا هي E. coli. Why E. coli is widely used in genetic engineering experiment due to short generation time for this bacteria. As you know, the generation time for the bacteria, which is the time required for one bacteria to be 2, is from 16 to 18 minutes. Thus, it is very suitable and is rapid growing microorganism and suitable for the use in the gene cloning experiment. After transformation of a recombinant plasmid, which contain DNA wanted to be reproduced, to be cloned to the bacteria, after that, the bacteria expose the third process, the third factor in this process. The bacteria will expose to the certain antibiotic. هنا هذه البكتيريا اللي هي نمت واللي بداخلها الريكومبينانت بلازميد اللي يحمل ال DNA wanted to be reproduced، راح نعرضها الانتيبيوتيك في داخل بليت، وهذا الانتيبيوتيك راح نختاره هو الاموكسيلين، لان قلنا احنا اخذنا اكزامبل، احنا نحمل هنا الجين المقاومه، هذا الجرينيش جين المقاومه للاموكسيلين، فحنعرض للاموكسيلين. هنا معيار انه انا عندي successful gene cloning experiment or failure due to growing of recombinant DNA. If the growing of bacteria which contain recombinant DNA will grow, that means the process is okay. Why؟ معناها انه انا الار جينز، ليش؟ لان معناها الار جين اللي يحمل الاموكسيلين ريزيستنس اللي يشفر الجين ريزيستنس، بالتالي راح يخلق المقاومه، لذلك البكتيريا راح تقاوم الاموكسيلين الموجود داخل الميديا، وبالتالي تنتعش وتنمو على سطح الطبق. في حين البكتيريا اللي هي اذا شفنا no growth on the plate، that is mean the process is failed، فشلت. ليش؟ لانه no transfer of a recombinant factor which contain resistance genes or gene encoding for amoxicillin resistance to inside bacteria. لذلك الاموكسيلين ما راح تظهر له مقاومه، فبالتالي الاموكسيلين يعمل انهبيشن للبكتيريا فما تظهر، فهذا معناها الفيلر for gene cloning. Okay, after obtaining now in success gene cloning, the bacteria containing recombinant DNA will grow and the culture and the conical flask and the manufacturers بالمصانع ونعملها DNA purification. Thus we obtain from one, we obtain 4 recombinant plasmid containing 4 DNA for gene need to be reproduced. فإذا احنا يعني قلنا انه احنا نعمل for reproduction of gene of insulin gene، هنا راح نحصل الانسولين بالعمليات بالصناعات الدوائيه من خلال الريكومبينانت فاكتور اللي يحمل الانسولين، ناخذه وينقون الدي ان اي ويسوون هارفستنج للجين ريبرودكشن فور الانسولين، وينتج الانسولين بالكميات الكافيه بالفارماسيز to treat the diabetes mellitus. Okay. Now, after I complete the first process, which is cell-based approach of gene cloning, I will talk on the second step or the second option of gene cloning, which is PCR. مثل ما تكلمنا، تقنية السيل-based approach هي الطريقة المعتمدة واللي في الفترة السابقة واللي كانت تستخدم بنجاح في عمليات الجين كلونينج، لكن نتيجة انه احنا عندنا دائما ما تتاثر بالتلوث البكتيري، وما يعاني كثيرا الباحثين وطلبة الدراسات العليا من التلوث اللي يسبب هو العامل الاكبر اللي يسبب الفشل. لذلك بعد اختراع البي سي ار باكاري ميولز بال 82 وحصول على النوبل برايس بال 93، البوليمر تشين رياكشن was introduced to the practice as the main process for a main technique for gene cloning. PCR, polymerase chain reaction, is defined as in vitro technique which allow the amplification of specific DNA region that lies between 2 regions of non-DNA sequence. A PCR amplification of DNA is achieved by using primer. What does it mean by primer? Here, if I have a patient, please to concentrate with me. If I have a patient suffering from TB and have the gene of TB, and as I know, the sequences for TB are 23 base pair, 23 nitrogen bases. This represents as a tablet, and primers consist of oligonucleotide which do as complementary to the non-template DNA of TB. يعني احنا ندز للشركة المصنعة نقول لهم هذا تسلسل اللي احنا نعرفه للتي بي، للفيرولنس فاكتور للتي بي اللي احنا اخذنا الجينات من NCBI National Center for Biotechnology Information. ندز للشركة المصنعة مثل شركة الألفا DNA الكندية، بروميجا الإسبانية، بلا بلا بلا، بايونير الكورية. فندز نقول نصنع البرايمر لهذه، صنعوا لنا برايمر او كومبلمنتري للتمبليت اللي انا اعرفه مسبقا اللي هو التي بي، لذلك نسميه برايمر او امبلايمرز. اذا these are short single strand DNA molecule which are complementary as I said previously to the end of defined sequence of the template اللي احنا نعرفه اللي هو التي بي مثل ما اخذناه كاكزامبل. The primers are extended on single stranded DNA by a DNA polymerase in the presence of deoxynucleoside triphosphate under suitable reaction condition. This results in the synthesis of a new DNA strand complementary to temperature strand. This strand exists at this stage as double stranded DNA molecule، لأن راح يكون عندنا التمبليت هو الدي ان اي المعروف مسبقا، والبرايمر اللي راح يكون عندنا كومبلمنتري، بالتالي يرتبط معه يصير الانيلينج فنحصل دبل ستراندد دي ان اي. Strand synthesis can be repeated by heat denaturation of the double stranded DNA, annealing of a primer by cooling the mixture, and primer extension by DNA polymerase enzyme. Then each cycle of DNA consists of denaturation, annealing, and extension. Each one cycle consists of these three processes, depending on the temperature. Denaturation, it occurs in 94 to 98 centigrade, depending on GC contains guanine cytosine ratio. طلابنا أود التذكير والتركيز هنا أنه كل ما كان البرايمر نسبة الجوانين إلى cytosine ratio بي أعلى، كل ما كان more rigid and not easily to be refractile. okay? لذلك يعتمد كل ما كان جي سي أعلى كل ما أحتاج درجة حرارة أعلى حتى أعمل له denaturation. وبالإضافة يعتمد على عامل ثاني هو long of DNA. كل ما كان تسلسل القواعد أطول كل ما أتوقع درجة الحرارة المطلوبة تكون أعلى. annealing بال 55 to 65 هذا البرايمر اللي أنا دزيت على الشركة صنعته وراح يجي ويعمل كومبلمنتري للتمبليت اللي أنا أساس عندي، يحتاج إلى certain temperature to anneal, to attach to the what, to the known sequences. هذه الحرارة تختلف من جين إلى آخر، كل جين أحبائي إلى درجة حرارة إلى درجة حرارة معينة، إلى برنامج معين بدرجات حرارية تقريبا شبه ثابتة فيما يخص الديناتوريشن والإكستنشن. تغيير بسيط يكون بالإكستنشن هو الثابت 72، ديناتوريشن رينج from 94 to 98، لكن المين ديفرنس يكون in the temperature of annealing. شوف some of genes 55, other 56, 58, 60, 65. يعتمد على annealing إذن temperature، يعتمد على melting temperature. إذن annealing temperature هو melting temperature 1-5. And the third step in each cycle is the extension 72. which is optimal temperature for tag polymerase. والتاج polymerase enzyme هو من اسمه تاج مشتق من الثرموفيليك بكتيريا أو نسميها الهايبر ثرموفيليك بكتيريا اللي هي الثرموس أكويتيكوس اللي هي تكون موجودة بالهوت سبرينجز يعني النبيع الحارة. لذلك قد نسأل ويسأل سائل يا دكتور مشتاق هذا التاج بوليمريز هو ممكن هو انزيم، هو اذا هو بروتين ان ناتشور، اذا مو المفروض يصير عندنا ديناتشوريشن درجه حراره 72؟ حنقول لا، السبب لانه احنا اساسا هذه الانزيم مشتق من بكتيريا متكيفه للعيش بالهوت سبرينجز درجات حراره عاليه، لذلك it is so difficult to denature by high temperature. اوكي، اذا كل سايكل، ايش سايكل عباره عن ديناتشوريشن الينج اكستنشن. لو ناخذ فد يعني تيبيكال بروجرام نشوفه، هذا البروجرام هو عباره عن التالي. اول ما نبدا نعمل انيشال ديناتشوريشن. الانيشال ديناتشوريشن هذا يخص الجهاز، هو لغرض الادابتيشن، يعني رفع درجه حراره الجهاز للعمل بدرجات الحراره العاليه اللي هي 95 سنتيغراد for 4 minutes. After that we will go directly to the cycle. راح نبدأ بالسايكلز، يعني ورا ما يشتغل لمدة 4 دقائق بالانيشال ديناتشوريشن راح يدخل بالسايكل. الفيرست سايكل هو عبارة عن ديناتشوريشن انيلينج اكستنشن. اوكي، ديناتشوريشن انيلينج اكستنشن هذه 3 خطوات. فالديناتشوريشن يحدث عادة اللي هو separation of double stranded DNA into 2 strands وبال 95 سنتيغراد for 1 minute. برايمر انيلينج مثل ما قلنا الانيلينج اوف برايمر اللي هو البرايمر اللي يعمل كتمبليت فور ككومبلمنتري فور ذا نون تمبليت دي ان اي بال 65 for example for this gene, while it differs from gene to others. The main difference in the program of PCR in primer annealing temperature. Okay, and followed by primary extension and 72, at which step the tag polymerase enzyme will add the nitrogen bases along to the reproduced DNA. After this will repeated for 29 times. يعني احنا راح يتكرر هنا عندنا اذا سايكل، بعدين يتكرر ب 29 سايكل مره ثانيه، يعني 30 دوره راح يكون بالمجموع. بعدين خارج هذا متى انتهى 30 دوره يعني ديناتشوريشن انيلينج اكستنشن ما يوحد فاينل اكستنشن، يستمر الجهاز ديناتشوريشن انيلينج اكستنشن ديناتشوريشن انيلينج اكستنشن for 30 times. After that it will go to the final extension. شنو يصير بالفاينل extension؟ بالفاينل extension that the same action or the same effect of tag polymerase, it will ensure the completeness of extension of all reproduced DNA fragments. And finally I will take an option to reach the temperature of the instruments to the refrigerator temperature اللي 4 سنتيغراد. ليش؟ to prevent renaturation. يعني احنا نترك الجهاز for example بدون هذه الخطوة اللي هي optional طبعا، لكن إذا كان عندك عمل ممكن أنت دا تصلي دا تاكل دا تشرب تحتاج 15 دقائق وربع ساعة، فتخلي الجهاز بالأربع درجة مئوية للوقت اللي أنت تحتاجه. تخليه ليش؟ حتى لأنه إذا الجهاز انتهى الجهاز وانتهت هذه الخطوة وبقى بدرجة حرارة عالية ممكن بيصير ريناتوريشن ديناتوريشن ريناتوريشن، يعني يرجع يلتصق مرة ثانية. لذلك to prevent renaturation we must reach to the hot temperature for certain degree and for a certain time as I needed. Okay. Then this represents the temperature profile for PCR. This represents the denaturation and more than 90 centigrade, followed by aliening and the 50 to 65 centigrade, followed by increasing to the temperature to 72 to do what? To do extension. Then denaturation, annealing, extension. Second cycle, denaturation, annealing, and extension, and also as we until you are reached to the final of the final cycle of the program, followed by final extension and hot temperature to complete the temperature of the PCR. In amplification phase, I have two phases, exponential phase and Plato phase. Exponential phase, this will be this lasts for all the time, for along the time for cycles. For example, if I have 30 cycles for a certain program, the exponential phase will extend along to the 30 cycle under standard reaction. After that, it will reach to the plateau phase. After completing the cycles inside the instrument of PCR, it reaches the plateau phase. In the plateau phase, it results from limiting amount of enzyme and reduce enzyme activity. هل ممكن يا دكتور مشتاق أنه نستمر بالسايكلز وبالبي سي ار إلى فترات أطول؟ لا طبعا، هذه البرامج اللي وجدت هي محسوبة بقياسا بكمية النيتروجين بيسز والاكتيفيتي اوف انزيم. لذلك من نصب البليتو فيز حيصير انه صار انه النيتروجين بيسز will be reduced in numbers, will be not enough for another cycle numbers. In addition to that, the tag polymerase enzyme will be reducing in activity. Okay, thus I will reach here to the plateau phase. Exponential phase and plateau phase, these are two amplification phase of PCR. إذن PCR programment هو البروجرام of PCR هو عبارة عن cycles, thermal cycles. وهي thermal cycles هي بها three main steps: denaturation, annealing, and extension, each of which has certain to do. Finally, in this lecture, I will, after we have take a brief introduction on the zero information of molecular biology and going to the gene cloning and taking the gene cloning process, either cell-based approach or PCR, in more details. Now I will talk on what is the medical importance of genetic engineering in medicine. The genetic engineering and application in medicine as follows. First, to produce large amount of biologically useful protein. These include A. Recombinant human protein used as a drug or hormones, like the following. For example, chorionic gonadotropin, HCG, it is used in the treatment of infertility. Also, it can be used for the production of insulin, which is very, very important in the treatment of diabetes mellitus. Interleukins, for the treatment of cancer, for the production of interferons, beta and gamma, in the treatment of viral infection and cancer. Also, for the production of growth hormones, in the treatment of growth retardation. Also, for a recombinant enzyme with industrial uses, renine in cheese making, protease in detergent, lipase in cheese making, in addition to catalase as antioxidant. The second purpose for gene cloning is to create organism that is especially in the sciences, in the College of Sciences and other branches of College of Science and plant, animal, and microorganism, which alter characteristic. For example, plant are resistant to disease. It is very important in the third stage and the third purpose in the diagnosis of most medically important bacteria. For example, tubercle bacilli, Mycobacterium tuberculosis, which is diagnosed with the technique of PCR, which is highly sensitive and specific. As you know, the conventional diagnosis of Mycobacterium tuberculosis in the culture may be extend to 1 month or more than 1 month. Thus, I need to use PCR for the production of specific gene, reproduction of specific gene encoded for Mycobacterium tuberculosis, and we can give you an cut-off diagnostic test for the Mycobacterium tuberculosis within 1 to 2 hours. Please, to show this difference in time between 1 month in the conventional diagnosis, while we can give the differential and cut-off diagnostic result for Mycobacterium tuberculosis within 1 hour. Fourth, for understanding the molecular basis of number of diseases. especially الفاميليال ديسيز مثل الفاميليال هايبوكوليستروليميا، يعني عائله كلهم عندهم الهايبوكوليستروليميا، العائله عندهم سيكل سيل ديسيز، ثالاسيميا، cystic fibrosis. All of these can be understanding by the molecular basis by gene cloning. Fifth, for the production of a protein for vaccination, for example, vaccine for hepatitis B, surface antigen, hepatitis B, and for diagnosis in the AIDS. And the sixth and the last purpose for the use of gene therapy in medicine and the application of genetic engineering in medicine in the gene therapy of sickle cell disease, thalassemia, and other diseases. All of these are application of genetic engineering in medicine. Till now the culture is complete. Dear students, hope to be interesting in this very important lecture in the field of molecular biology, and please do not hesitate if you have any inquiries or any questions to call me directly by sounded message or by video message or by Google Classroom and through the all communication tools. I am ready for answering all your questions and queries. Thank you very much for your patience and for giving me a time for giving a lecture, a video lecture in the gene cloning. Thank you very much. Hope to be success. Thank you.